Okay, before we begin, are there any questions? Here. Oh, I need to open up chapter 8 also. Let me state that today I'm gonna give you guys a break at 10 minutes to 6. I've got something I need to take care of at that time. I hope the break will only be 5 minutes. I'm certain it won't be more than 10 minutes, but we will take at least a 5-minute break at 10 minutes before 6. Any questions about any of that? There's no questions. Let me share my screen. Forgot they changed that. There we go. Now if that'll go away so I can get this down and out of here. We'll come back to that. All right, can somebody confirm you can hear me?
Yes.
Thank you. All right. We're supposed to be working on chapter 14 today. We're still on chapter 8. We are behind schedule. For those in the lab, 7 p.m. lab, I'm going to show up for the first 15 minutes to answer questions, but there is no lab today. Because we're behind schedule, I might move this review to the lab schedule. However, at the moment, I'm hoping I can catch up and we'll keep to this schedule. I'll let you know. I'll post an announcement if I change the review schedule. The review is a review for the final. All right, let's go to chapter 8. And that won't be right because that'll be the Monday, Wednesday, but let me find where we are. Oops, that's the wrong one. Oh, still the wrong one. There we go. I don't remember. I think you guys are actually behind schedule. A little behind Monday, Wednesday. All right, so I talked about the complication that RNA processing of messenger RNA in eukaryote cells involves removing the exons from the original RNA transcript. And only when removing the exon, removing the introns, sorry. And only when the introns are removed can that final messenger RNA leave the nucleus to go into the cytoplasm to be translated. So we talked about the structure and function of the genetic material. Let's now talk about the regulation of bacterial gene expression. We'll talk about operons. We'll then switch topics and talk about mutation, genetic transfer and recombination, and lastly, genes and evolution. Have we talked about
Yeah, we've talked about lac operons and and mutations.
all right, so that's what I was thinking. I didn't think you guys were that far behind.
I
If
think
I was
we
that far
only
behind.
had like 10 slides left.
Yeah, I must have not removed that. Let me try this again. There we go. There we are. So we had talked about transformation, where a cell can pick up a naked piece of DNA and incorporate it into its chromosome and express it. And this is one way that genes can pick up pre-existing genetic information, horizontal gene transfer. The first way is transformation. We're going to talk about conjugation and transduction. These are all the 3 ways that cells can pick up pre-existing genetic information. Now, if it isn't pre-existing, they can't get it by transformation, conjugation, and transduction. The only way they can get it if it isn't pre-existing, is by spontaneous mutation. This is a cell picking up a piece of DNA, consuming it, getting some indigestion, and then changing its phenotype when it expresses the genes on the DNA. The second way that cells can pick up pre-existing genetic information is known as conjugation. This can happen in two ways, depending if the cell is a gram-negative bacteria or a gram-positive bacteria. We'll mostly talk about it with the gram-negative bacteria because, well, they have special structures for doing it and it's been studied a lot. The gram-positive way, we'll talk about it first, is two gram-positive cells can come together and then the cell wall dissipates in this region here so that the cell membranes can touch each other. The cell membranes can then fuse with each other so that the cell membrane, instead of coming across like that, is now going to go here and then around here and around there and then around there. And that means the cytoplasm of this cell is connected to the cytoplasm of that cell. A plasmid can move from this cell here into that cell or vice versa. And that is how conjugation happens in a gram-positive cell. This arrangement is called the mating bridge. And it does happen in gram-positive cells. Any question about that? All right. In gram-negative cells, it happens by this cell, which is an F plus positive cell, meaning F positive cell. It has an F plasmid in it. The F plasmid codes for the construction of the sex pilus, this structure here. The sex pilus will go out and literally skewer an F minus cell. And then the sex pilus can then act as a winch and pull the two cells closer together. They do not have to be this close together. They can be, oops, didn't get that right. They can be this far apart because that sex pilus is hollow. And what happens is the F plasmid can replicate, make a copy of itself, and then send a copy across the sex pilus into the F minus cell. That F minus cell now has the F plasmid. The F plasmid codes for this cell to grow a sex pilus, and then this F minus cell will become an F plus cell. It'll go out and skewer an F minus cell and then transfer a plasmid, the F plasmid, into that F minus cell.
So does F minus mean that it just doesn't have a plasmid in it?
It doesn't have a plasmid in it, and it doesn't have a sex pilus either.
Got it. Thank you.
Okay. And when I'm talking about it doesn't have a plasma, I'm talking about the F plasmid coding for the sex pilus. Okay. And if you notice, there are a few other differences between these two cells. That cell is rounder than that cell. This one, not only does it have the sex pilus, it has a bunch of, what are those called? Fimbriae on it. And that one, it might have some short fimbriae right there, but certainly nowhere near as many as that one. The third way that pre-existing genetic information can move around in a population of cells is transduction. Transduction involves a virus. We haven't talked about viruses yet, so let me introduce a little bit about viruses. And that is, a virus can bind to this cell and then inject its genome into the cell. The genome of the virus, the first thing it does is it codes for the destruction of the chromosome to this cell. The reason why the virus wants to do that is the virus genome wants to call the shots in this cell. And what it's going to want to do is replicate itself in the cell. And one way to call all the shots in the cell is to destroy the host chromosome. And so it makes a, what is that called? Nucleases, which degrade the host chromosome. And then the viral genome replicates itself and then transcribes to make viral proteins. The viral genome and the viral proteins get together to make the new viruses. And then the virus lyses the cell to release the newly replicated viruses. And that's the way viruses, or at least some viruses, some viruses live. Let's just word it that way. It's actually called the life cycle, but we will get into that in a later lesson. So the virus does that. But let's take a look at something right here. The viral genome is this reddish purple. The host genome is in pieces, and it's the darker purple, blue purple. And the virus occasionally makes a mistake. And viruses aren't terribly worried about the mistakes they make because they have lots of offspring. And if one of them isn't quite correct, it's not a big problem to the virus. These are correctly replicated viruses, but take a look at that one. That doesn't have the viral genome in it. What was stuffed into the virus was a piece of the host DNA, host chromosome. And now when this virus infects a cell, the virus will bind to the cell in the normal way, and it'll inject its genome into the virus. But this is not the viral genome. It's a piece of this chromosome, to this cell's chromosome. And then that chromosome can recombine with the, let me shrink that down. There we go. Can recombine with the host chromosome and then be expressed. So that these genes are genes that were not in this cell. They were genes that were in this cell. And an important part of this is this, if this cell is a cell that has a gene for antibiotic resistance, that gene can move into this cell by this virus. And then the cell can express the gene for antibiotic resistance. Any question about any of that? This is called transduction, involving a virus. Transduction probably happens in human cells in the nose and maybe in the throat all the time, but it only will affect the DNA of a nose cell or a throat cell. It will not affect the DNA of a germplasm cell, so it doesn't have any effect on the human population. Any question about any of that? All right, so those are the three ways that pre-existing genetic information can be spread around in a population. The importance of all of them, let me make a new file here, make this big so it's nice and easy to see, is, come on, that spontaneous, why didn't that write? I'm going to abbreviate that. If the genetic information does not occur in the population, there's no way it can spread around. The only way it can come into being is by mutation. And mutation will happen about 1 time in 10 to the 6. Oops, shoot, got the wrong one there. 1 time in 10 to the 6 cells or genes. We'll just say cells for this, making it easy. Oh, come on. I did a space there. Where is it? There it is. Transformation, conjugation, and transduction. I'm going to just write it in one place. So let's go ahead and let me see if I can add a symbol here. Probably can't. I will just do it here. It's from all three of these. I'm going to be a little speedy here and only do it one time. Transformation, conjugation, and transduction in at least gram-negative cells, maybe gram-positives as well, will happen about 1 in 10 to the 5th cells, meaning that they will happen 10 times more frequently than spontaneous mutation. So if we have a cell that gets a new phenotype, such as antibiotic resistance, it is most likely to come about from a pre-existing cell by transformation, conjugation, or transduction. It can come about by spontaneous mutation, but it will happen 10 times less frequently than a new cell getting that gene by transformation, conjugation, or transduction. Any question about any of that? All right. So let's talk a little bit about plasmids. For one reason, we need to talk about plasmids because a cell is more likely to exchange a plasmid than it is a piece of its DNA from the chromosome. So plasmids are important to cells, especially for cells engaging in sexual conjugation, but also in other means, like transformation. A plasmid is more likely to come out of the cell and then go in the environment and then be picked up by another cell that will be the recipient. Plasmids differ from the chromosome mainly in their size. Plasmids are small, circular, extrachromosomal pieces of DNA. They only contain a few genes, as shown in this chromosome, having maybe 1, 2, 3, 4, maybe 5 genes, and I might be missing some because I don't know what these are standing for. So it might be as many as 8 genes, but only a few. Whereas a chromosome has tens of thousands of genes on it. If we're talking about E. coli, it probably has 20,000 genes on it. So the chromosome is much larger, but they both are circular circular molecules of DNA. So in that sense, they're the same. The plasmids can replicate independently of the cell's chromosome. And I actually showed you that before when we were talking about the F plasmid replicating. Where is it? Right here. That plasmid replicated, went into that cell. The chromosome did not replicate. So that plasmid was replicating independently of the chromosome. And plasmids can do that. Any question about plasmids? There are different categories of plasmids. You don't need to know the names of the categories or what they do. You just need to know that there is more than one type of plasmid. Let's talk a little bit about transposons. Transposons are an odd piece of DNA in that it's a piece of DNA that moves about in the DNA. So this piece of DNA can excise itself from its location and then move and insert itself into a new location in bacteria in the same chromosome. But well, we don't know about transposons. I don't know if they know about transposons in humans. In eukaryotes, the transposon can move from one chromosome into another chromosome. And it actually excises itself from the first chromosome and then moves into the second chromosome. So there are segments of DNA that can move about from one region of DNA to another. Transposons always contain the transposase gene. That enzyme will cut out the transposon, and then in the new part of DNA, it will cut that DNA and then insert the transposon into that cut first portion of DNA. Transposons contain, of course, the transposase gene, and they also contain an inverted repeat on the sides or the edges of the transposon. It's thought the inverted repeat helps the transposon insert into a new location of DNA. Any question about that? So that's a simple transposon. All it has is the transposase gene and the inverted repeats on both sides of the transposase gene. You can have a complex transposon that carries other genes on the transposon, such as this transposon here, transposon number 5, that is carrying the kanamycin resistance gene. And this complex transposon usually has the other gene of interest, in this case, kanamycin resistance. And then it has a transposon over here and a transposon over there, meaning this will be coding for the transposase gene, and so is that. And there's the first inverted repeat. There's the other inverted repeat. There's one inverted repeat. There's another inverted repeat. Any question about any of that?
What's
Transpo-
the
Go on.
what's the purpose of the non-complex transposons?
Purpose. You're saying why are they in a cell?
Yeah, what does it matter if it's not carrying other genes with it?
Like I said, it's just a part of the genome. Why it exists, you have to ask God or the universe or whatever you want to ask your question to, because I'm not sure if they know why it exists. Obviously, it can move around DNA,
Okay.
okay? But why does a cell want to move around? Like the kanamycin resistant gene is perfectly good in its original location. Both places it's going to code for kanamycin resistance. Okay, so I'm not sure how to answer your question because I'm not sure if anyone even knows.
Okay, that's fine,
Right?
thanks.
We've known about transposons since at least the 1960s, and they actually first discovered it in corn. Somebody was trying to map where the gene was, where the corn kernels were changing, and they discovered that the location of that gene moved around. Okay, so that was one of the first cases of transposons. And then they discovered them shortly thereafter, I think in E. coli or some prokaryote cell. And they realized that there's regions of our DNA that actually move around. And I'm not really current on the transposon literature. When I was an undergraduate, there were no transposons known in the human genome. But I don't know if that's still true because that was a long time ago. And obviously that was before we sequenced the human genome. Now that we sequenced the human genome, they would know if there's transposons in the human genome. And I just never looked it up, so I don't know. But there are transposons in eukaryote cells, so it's certainly possible that humans have transposons. Now, for bacteria, it does allow the gene, like kanamycin resistance, to move from one location to another, such as move from a chromosome and into a plasmid. Why this is important is that plasmids are much more likely to be shared between different cells. So a plasmid is more likely to be inserted into a virus, and then transduction can carry it into another cell, or a plasmid is more likely to undergo transformation. And certainly a plasmid is much more likely to undergo sexual conjugation than the chromosome. So that's the importance of transposons. They can move about from one location to another, including move into a plasmid, and then that would increase the chance that this kanamycin resistance gene would then move into another cell by some means like sexual conjugation. Any question about any of that? All right, well, transposons were just something I needed to mention because it is some part of genetics, and I don't know if any of you have ever heard of transposons before, but they are regions of DNA that move about in the genome from one location to another. If not, let's talk about genes and evolution. It's a real quick one slide. Genetic variation, if it is new genetic variation that first appears in the population, it has to come about from mutation. An example of that was the blue-eyed allele occurring in Northern Europe about 10,000 years ago. It resulted from mutation. And that's the same thing with the first variant that arises in a bacteria population as well. It has to come about by mutation. But once it exists, crossing over can reshuffle it and express it with other genes. That's the purpose of crossing over, just making new combinations of genes. And then transformation, conjugation, and transduction can spread that new variation around in the population. And transposition can help the spread by moving that gene from one location in the genome to another, especially if it moves it from the chromosome to a plasmid. All of this genetic variation then drives evolution, and evolution will proceed by natural selection. Any question about any of that? All right, so that's genes and evolution. That's it for chapter 8. Any questions about chapter 8? Before I close it down, and I guess I should save that. And then we'll open up the next lesson, which is chapter 14. The last, well, second to the last lesson will be chapter 13 on viruses. So let's quickly move through chapter 14. This, what the heck? Oh, I've still got that up here. That's what the trouble is. Chapter 14 is Principles of Disease and Epidemiology. I've only got a few terms up here, but if you learn the terms of this lesson, you will learn about half of the chapter. So you do need to learn the terms of this chapter. They're very important for this chapter. We will also talk about the relationship of the normal microbiota and its host. You do need to understand the stages of disease development. And then lastly, understand what nosocomial infections are and how to control them. Boy, I think in the lab I mentioned about nosocomial infections. I didn't realize we hadn't discussed it yet. My apologies. All right. In the principles of disease and epidemiology, the first three terms you need to know are pathology, the study of disease, etiology, the study of the cause of a disease. For example, the etiological agent of the disease anthrax is Bacillus anthracis. Sorry, I was trying to remember the name of that. Bacillus anthracis causes the disease anthrax. Pathogenesis is the development of a disease. Infection is the colonization of the body by a pathogen, or it could be the presence of a microbe where that microbe is not normally found, such as E. coli. If it gets in the lungs, E. coli should not be in the lungs. It is a part of our normal microbiota, but if it grows in the lungs, it would cause pneumonia, and that is an infection as well. A disease is an abnormal state in which the body is not functioning normally. When we're talking about disease and microorganisms, you should realize that very few microorganisms are pathogenic. Very few microbes cause disease. Some can even benefit the host, especially those that are part of the host's normal microbiota. Any questions about any of that? All right. Each animal and plant has a normal microbiota, a large community of microbes found in association with the animal and plant. For humans, you should call it the human normal microbiota, because even of mammals, for example, they will have different normal microbiota. Like, for example, cats have Bartonella henselae as part of their normal microbiota. In humans, if we have that, it is only during an infection, and it causes cat scratch fever in humans. Any question about any of that? So humans have a normal microbiota, and so do cats, and obviously any other animal. And there may be overlap, like between cats and people. There's a large number of organisms which are similar, but there are differences too. The community is known as the normal microbiota. And when you speak about the human normal microbiota, you should use the term human normal microbiota. Although if you use normal microbiota, people will probably assume you're meaning the human normal microbiota. The normal microbiota are microbes that permanently or close to it colonize the host and then remain with the host for essentially all of their lifetime. The normal microbiota is only found in certain areas of the host. Disruption of the normal microbiota and the host balance appears to be associated with disease and with impaired immune function. One way to disrupt your normal microbiota is to take antibiotics, especially orally. That will disrupt your gut normal microbiota, as well as your normal microbiota and the rest of your body as well, but especially your gut. And that will frequently cause, not always, but can frequently cause either constipation or diarrhea. I don't know about you, but sometimes when I've taken antibiotics, I've gotten diarrhea. I don't think I've ever gotten constipation, but I have had diarrhea. And it doesn't always happen, but it just frequently happens. Unfortunately, the normal microbiota host balance reestablishes And then after usually a day or two, the diarrhea goes away. Any question about any of that? All right, this map is picture is trying to show you some of the regions where the normal microbiota are found in the human body, and that's obviously all of the skin, all of the mucous membranes open to the outside, like the nose, the throat, the eyes, the mouth, the large intestine, and obviously the gastrointestinal tract has normal microbiota. The urinary and reproductive systems, at least the lower portion, it does have a normal microbiota. The urethra on both males and females has a normal microbiota. Any questions about that? Places where the normal microbiota are not normally found include the internal structures and organs like the blood, the cerebrospinal fluid, the muscle, nervous tissue, bone, heart, etc. Any organ that's inside and not open to the outside. The normal microbiota differs from the transient microbiota in that the normal microbiota normally, once it gets established, it will colonize the host for essentially the entire host lifespan, like Staphylococcus epidermidis. Humans get that shortly after their birth, or maybe even during their birth, because I'm sure you get Staphylococcus epidermidis from the vagina. And then that Staphylococcus epidermidis, once it colonizes the baby, it will last with the baby almost the baby's entire lifetime, if not the baby's lifetime. Staphylococcus epidermidis probably will stay on the baby for the baby's lifetime. Transient microbiota, on the other hand, may be present for a short time, days, weeks, or months. Unlike the normal microbiota, it does not permanently get established on the host. I once had a transient microbiota, and this is back when I was working at another lab. I would routinely, each term, sample my skin for Staphylococcus aureus, and I never had Staphylococcus aureus on my skin, and I sampled my skin for years. And then I went to visit my aunt, and she had a Staphylococcus aureus skin infection, and she was taking antibiotics, so she probably assumed that she was not contagious. But when she saw me, she insisted on hugging me and my mother. I took my mother to see her sister. And I happened to sample my skin just a few days after that hug. And for the first time, I had Staphylococcus aureus show up on my skin. And I said, geez, I must have picked this up from my aunt because I never had it before. And I was kind of worried because Staphylococcus aureus, if you do not know, is a human pathogen, although 20% of Americans have it on their skin. And it's close to 40 to 50% of Americans have it in their nasal passages. But of the 20% of the Americans who have it on their skin, they're much more likely to get boils and other skin infections from Staphylococcus aureus. Anyways, I then tested myself the next term, and I no longer had Staphylococcus aureus. And I was relieved, saying, whew, it was a transient microbe. It was on my skin for a short while after I hugged my aunt. And I'm sure that's where I got it. And fortunately, my normal microbiota took over and threw off Staphylococcus aureus. And so I no longer had it. And I sampled my skin for each term for maybe twice a term even, initially, for a long time to make sure I never had Staphylococcus aureus again. So for me, that was a transient microbiota. Any questions about any of that? The normal microbiota has recently been found to have 4 times 10 to the 13th bacterial cells on the human. The human body themselves have only 3 times 10 to the 13th human body cells, meaning there's more bacteria cells on us than we have our own cells. And that begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much. It's just part of our normal microbiota. Any question about any of that? Here's some pictures of the normal microbiota in the nasal epithelia. This is the cilia of the nasal epithelia, and the cocci are the normal bacteria. There's some normal bacteria in the lining of the stomach. That right there is part of the stomach, and that's the villi in the stomach. Here is some bacteria in the large intestine. Right there, you can see some food being digested, but there's well, maybe a rod. I'm not sure what that is, actually. There's certainly a rod. These are coccobacilli. That one might be even a cocci. The human normal microbiota has been found in some recent places, meaning fairly recent. It's how many years ago was that now? 10 years ago. They found that the normal microbiota has been in some surprising places, like the female urine in the bladder. Females have normal microbiota in the bladder. It was assumed before this time that the bladder was sterile, but at least females, they have a normal microbiota in the bladder. And I've looked, nobody's ever done research on males, so I don't know if males have normal microbiota in their bladder or not, but females at least do. Any question about that? Another case which is kind of, I don't know, odd, is that the mother's milk is not sterile. Mother's milk, even if you take it out of the breast directly, has a normal microbiota in it. There is bacteria in the milk inside of the mother. Now, when the baby is nursing, obviously the baby is going to get the bacteria from the mother's nipple. However, it turns out that even if we were to sterilize the nipple, which is kind of hard to do, there would be bacteria in the milk that the baby would drink. And so that's a surprising place. We thought that mother's milk was sterile. I'm sure that's the case with cow's milk too. There must be normal microbiota in the cow's milk. And they assumed previously it just came from the nipple. And I don't know why this is a surprise, but they've discovered that the placenta of a pregnant female has a normal microbiota. And why this is not a surprise to me is the female reproductive system is open to the outside. So it's not surprising that the bacteria can come in and come up to the placenta. What is unique is the placenta has a unique microbiota that is unique to the placenta, meaning this microbiota is found nowhere else in the human body. Any question about that? All right. When we're talking about the normal microbiota, it's important to realize it establishes a relationship with the host so that there is a balance between the host and the normal microbiota. The normal microbiota can also compete with pathogens. Microbial antagonism is the competition between two species of microbes. The normal microbiota can compete with human pathogens by occupying the niche that the pathogen might try to occupy. For example, I've got lots of Staphylococcus epidermidis on my skin. If Staphylococcus aureus were to fall onto my skin, it might fall on top of Staphylococcus epidermidis, and then the Staphylococcus aureus cannot get established. So it can occupy the niche. They can also compete for resources. If Staphylococcus epidermidis use up all the resources on that skin, and Staphylococcus aureus were to fall nearby on the skin and not have any resources, the Staphylococcus aureus is not going to get established. The normal microbiota can also change the environment, making it less hospitable for the pathogen. Frequently, the normal microbiota can produce acids, such as in the urethra or the vagina, or even in the mouth. And that can make the environment too acidic for a pathogen. Another case would be the normal microbiota makes your skin more acidic. Sebum is slightly acidic. I don't remember what the pH is, but the normal microbiota metabolizes the sebum and makes it a little more acidic. So the skin too is an acid. Bacteriocin can be produced by the normal microbiota, and bacteriocin is a protein that inhibits the growth of a certain class of microbes. Like E. coli, if it's producing a bacteriocin, that E. coli will prevent, or the bacteriocin from E. coli will prevent the growth of Salmonella and Shigella. So a bacteriocin prevents the growth of similar bacteria to the microbe which is making the bacteriocin. Sorry, I had an itch on my nose there. I've often wondered why bacteriocins are not used as an antibiotic, because if we were to use bacteriocin from E. coli, you could cure a Salmonella or Shigella infection, and it would not harm other bacteria like Enterococcus faecalis in your gut. Any question about that? Bacteriocins are being experimented with to use it as an antibiotic. I read about that a few years ago. I'm not sure how far along the bacteriocin research is, but they are experimentally used now for treatment. And then lastly, probiotics, the use of live microbes intended to exert a beneficial effect. You can buy probiotics almost anywhere now. Kimchi and yogurt have live, if it's kimchi or yogurt that have live cultures in it, that is a probiotic. And if you consume yogurt that has live bacteria in it, you're much less likely to get a yeast infection. All right, any questions there? If not, it's time for me to take a break. I hope to only be for 5 minutes, but certainly no longer than 10 minutes. I will stop the share and stop recording